Take the blood test, you have a given set of features, but the same features are measured to millions of other patients, okay? So you have populations, and the idea is, can I find a representation which quantifies the deviation, the difference of a given subject with respect to a given class? So imagine to have a labeled class, which can be healthy, non-healthy, can be with a given disease, without a given disease, or whatever class you can imagine. I have many individuals of this class, so I want to extract, learn from this class something, and I want to associate then a network to a subject, to an unlabeled subject, subject without label, to tell how much this subject is distinct from a given class. So this is the concept of parenclitic networks. And the way you construct is rather simple. So you say, suppose that we have n people, n system, or n subject. These are all the people who goes and make the blood test. And these people is associated, these are the labeled people, because they are associated to one of M classes, C1, Cm. In the simplest case, the class can be associated to only two classes, control people, healthy and sick, okay? Okay, so I have these data sets, and each people, each person, each system is identified by a vector of p features. So these are the p features that I measure when I do my blood test. Which means that each one of these persons is a point in a p-dimensional space. So I have these persons, and each one of them is a point in Rp. Okay? This is Rp. It's not R3, it's Rp. Maybe many of such features. Now, the ansatz you make, the hypothesis, is that each class, means being healthy, being control, being sick, being whatever, correspond to a set of constraints in the feature space. What does it mean? It doesn't mean that I take my space and I project this space in all possible plane in which I have feature I versus feature j. Okay? Here I have all features and I have lot of persons. Here I have all possible planes in which I can project this space, in which I have, for example, here I have feature P against feature 1. Here I have feature 1 against feature 2. And here I have, in general, feature I against feature j. In each of these planes, my population, my class, will correspond to a given number of points. Okay? Now, what my hypothesis is that for each one of these planes, there is a function, a constraint, Fij of Fi, Fj equal to 0. So these points are not distributed in a cloud, but they are distributed along a given function. This is function 1, this is function in this space, this is function in this other plane. So each plane has a specific constraint. Notice that this is not a function, it's a constraint. This does not mean that Fp is function of F1, but a given function of fp f1 is equal to 0. Okay? So it's a constraint. So I imagine that each class corresponds to these constraints, and if I have good methods, like polynomial fit, data mining methods, support vector machine, artificial neural network, I can learn these functions. From my labeled set, I learn all the functions. Correct? And now an unlabeled subject comes, which is me. Being an unlabeled subject is an extra point here, and an extra point here, here, and here. In all of this plane, the new subject is an extra point. And so I associate to this black person one network where the nodes are the fissures. So this is fissure 1, this is fissure 2, this is fissure 3, this is fissure 4. And the links, for example, to define the links between fissure 1 and fissure 2, I go to the corresponding plane, fissure 1, fissure 2, and I measure the distance of this point to the function, to the constraint. This distance will be the value that I put here, dij, d12. To define fissure 1 with fissure n, I go here, and the distance will be this one. To define fissure the link between node I and node j, I calculate this distance. Okay? It's clear? This is the same I made on the whiteboard is made here. So I have many subjects. Each subject has many fissures. Each person corresponds to a group of points in the fissure space. Here you see you have two classes. One class which are the green points, and another class which are the blue points. Then you go in each one of the projected planes, okay, and the blue points will be defining a function, and the green points will be defining another function. So I will have the set of functions defining class green and the set of functions defining class blue. And then when a new subject is arriving, this new subject is a red point. This red point is associated to a network in which the nodes are the fissure, you see, and the link is nothing but the distance between the red point and the class with respect to which I am calculating the network. That's why it's called a parenclitic, because each link quantifies the distance of a given subject from a given class. So this is a construction which is defined uniquely. And notice that this construction is in fact a multi-layer network, because this link is not, if you have more than one class, this link is not a value but is a vector. The first component is this distance, the second component is this other distance. Then if you have a third class, for example, the gray point, you have another set of constraints corresponding to the third class, which defines you a third component of the vector, and so on and so forth. But now suppose that you have only one class, the simplest case. So suppose that there are only the green people, which, for example, are the healthy people. Then with only the green people, you calculate the green functions, and you have a network in which this is one component, so it's the distance of between the point and the green functions. So Applications, typical applications are in biomedicine. Why? Because you have this kind of data which are expression of fissures. For example, this is an application on early diagnosis of obstructive nephropathy. This is a disease which is particularly dangerous in newborns because it's the primary cause of kidney transplant in newborns. And this is a disease in which the urinary tract is blocked, so part of the urine is shut back to the, is sent back to the kidney, and if you don't treat this urinary blockage, the kidney can be damaged seriously, up to a point in which you need transplant. You need a kidney transplant. Here what you can do is you can measure the urine of the babies, and you can measure the concentration in the urine of 852 metabolites and 834 regulators. And you have 10 patients having obstructive nephropathy and 10 patients which form the control group. So the healthy people. So what you do, you take the healthy people and you learn all the functions on the healthy people. So you learn all these functions. Then you take the unlabeled subject. If the unlabeled subject is a healthy people, you see that your network is almost not structured. And this is clear why it's a sort of random network. And this is clear why, because you are comparing a member of the same class with other member of the same class. So the differences are somehow random. They are the fluctuations that you have in a class, in the population of a class. So you don't expect to have a very clear structure over here. Instead, if you take a patient and you compare this patient with the class of healthy subject, what you obtain is a network which is very well structured. It's a star. And the star means that not only this allows you to classify, because this is evidently different than this, so you can say you have the disease, but what is most important, you can individuate the specific feature that makes this specific subject different from the class, which is the center of the star. Why? Because since this is the center of the star, it means that in any plane in which this feature is one of the two axes, the distance of the person is very large. Whereas the fact that, for example, there is no link from this to this means that if I go to this plane, the subject is is not far away from the constraint. I forgot to tell you that when you calculate this, you obtain, like in the functional network, an all-to-all connected network, of course, because and then you have to threshold. So this means that as a result of the threshold, you have links in some subspace and no links in some other, which corresponds to those planes in which you are almost within the constraint. So this is very important because this is not only a classifier in the sense it allows to tell you you are belonging or not belonging to this class, but this is a personal classifier in the sense it tells you why you are not belonging to the class, which is the most important node feature that distinguishes you from the class. Then we apply to a second case using exactly this property to longitudinal evolution in time of some process, and the process is the reaction of a plant to osmotic stress. Why? Because at that time I was in Israel, and in Israel there is a big, huge problem, which is how to produce food. You know, Israel is living not in peace, so they want to be completely self-sustained in food and water. So they desalinize everything for the water, and they have to make plantation for food, for tomatoes, for potatoes, and the problem of Israel is 75% of the land is desertic. So the soil is very bad. There's no precipitation, and the soil is very acid and very salty. So they need to make OGM. They need to take a plant and make it resistant to conditions which usually this plant is not having. So the question is, sorry. Sto dando un seminario, non posso. Ti richiamo dopo. So the question is, I take a plant, which is the Arabidopsis thaliana. Why the Arabidopsis thaliana? Because the Arabidopsis thaliana is the plant for which I know exactly the entire genome. I give to this plant a osmotic stress, and then I measure the complete genetic expression levels at 5 minutes, 10 minutes, half an hour, 60 minutes, 120 and 240 minutes after I gave my stress to the plant. So I have 6 expressions of these genes. The genes are 22,591, and I measure this 22,591 level of expressions 6 times. What I do is at each time, 5 minutes, I take all the other times, all the other expressions are as normal. So I have at each time 5 other expressions, and I consider these 5 other expressions as normal behavior. I learn my functions on these 5 expressions, and I look the parenclitic network which distinguishes at 5 minutes my expression with the rest of configurations. It's clear what I'm doing. Then at 10 minutes I do the same. Now at 10 minutes I take 5 minutes, 30 minutes as a normal expression. I recalculate my constraints, I relearn my functions, and I construct a second parenclitic network at 10 minutes. Then I construct a third parenclitic network, 30 minutes, 60 minutes, 120, 240, and what I obtain generally is a collection of stars. Look at this gene, because now you look at this is a gigantic hub. Here is a gigantic hub. You see how many other genes have connections with this. Okay? So my interpretation is that this center of genes are indeed the most important and fundamental genes that play a role at different times in the response of the plant to the stress. Because is the way, the place where they are mostly different. And then what I do, I simply study these 6 parenclitic networks, and I study centrality, measure centrality, and I make the rank of centrality. So obviously the center of the ones which higher centrality are the center of the stars. So I take the first 5 genes at 5 minutes, the first 5 at 10 minutes, the first 5 at 30 minutes, the first 5 at 1 hour, 2 hours, 6 hours. And you see here you have the value of the centrality. And then with this list, I go to plant genomics and say I predict that these 30 are the most important genes that regulates the behavior of the plant in response to this kind of stress. Plant genomics said, I don't know. So we organized an experiment. And the experiment consisted in, we first grew one plant. This is the Arabidopsis thaliana. This is a photo. We grew a plant without touching anything, just giving the stress, the osmotic stress. And you see after 6 hours, the phenotype of the plant was such that the plant survived and developed this root length plus minus a given fluctuations, because we made, of course, 100 plants per experiment. And then we constructed several transgenic lines in which we blocked the expression of a specific gene. This is a very tiny, small perturbation because you have 22,000 genes, so the transgenic lines is blocked only one gene. All the rest of 22,000 can do whatever they want, but that specific gene cannot be expressed. There are techniques that are called kickoff. You take out this gene. And of course, what you take out is one by one these genes. You take these genes, the one you predicted, have an important role in the plant, and you avoid the plant to have expression of these genes. When you do this, you see that in all cases, for all genes that you predicted, the phenotypes is completely different. In some cases, for this gene, for example, the plant almost dies. For other, it survives, but the root length is very low, much below the normal, the wild phenotype. In some other case, it even overshot, so it grows too much. So really, in no cases, you see here the error, you stay in the population of the wild phenotype. So it means that if you touch indeed one by one these genes that you determine a completely different phenotype of the plant. And as a counterexample, what we did is taking instead two genes which are peripheric, like these two, touching these two genes, and demonstrated that in this case the plant grows with exactly the same phenotype as the wild type. So it means that really these are the genes which are the most important. So this was a very long experiment because it took us many transgenic lines, very costly, but it was a demonstration that, for example, alpha centrality in this case of networks gives you information, very precious and important information. We are applying this to other cases. We have a large project in Madrid on application to functional magnetic resonance data of patients of Alzheimer. And here we use the full multi-layer description because we have several classes. We have control subject, which is subject which are good, but then we have different levels of Alzheimer, and therefore we can discuss how a given subject is positioned within the typical evolution of Alzheimer's disease that goes from the mildest case, which are the amnesic mild cognitive impairments, to the conclamated Alzheimer. So you can really tell a person how is positioned into the normal course of the Alzheimer's disease, or at least what we know of the normal course of Alzheimer's disease. But you can understand that in principle the method has potential applications to all cases of data in which the data have no time evolution and are basically static expression of features. So this is a series of other application of the method which was done by other people. They applied for cancer, basically cancer identification, credit card fraud detection, prediction of ovarian cancer, mild cognitive impairment, and recently to cybersecurity. So this is a method of using network representation of this data which is useful, and certainly it can be seen as an extra method you can use in data analysis. Okay, so I don't speak about this. I think I gave you sufficient information on networks. I give you, however, this is important, a few literature, because you know I had to condensate in 4 or 5 hours a lot of concepts and a lot of different things about network structure, network dynamic, application of network for data. And this is just to give you a flavor of complex networks and what they can do. If you are interested in some specific part of these classes, or you want to deepen your understanding on some problems, you can read 3 important monographies. Actually 4 important monographies. One is this paper of 2006. It's Complex Network Structure and Dynamics, where you find, for example, the first version ever of the master stability function, and you find some other general description of network. This is a very huge, more than 100-page monography on networks with 888 references. So it's a huge, so to say, review. Then we made the same, but now applied to multi-layer networks. I didn't speak about multi-layer networks, but multi-layer networks are those networks in which you have different layers of interactions. And if you are interested in this, this was published in 2014. Again, is almost 100-page of review with more than 500-600 references on networks. Then, if you are interested to network with data, this is the first reports that we wrote on 2014 again, which tries to compare methods from data mining with methods of networks, so functional networks and parenclitic networks. So there is a list of a combination of these methods and, in general, algorithm for data analysis. And finally, if you are interested in synchronization and other processes, the very last one is a huge report we wrote in 2016 on explosive transitions in complex networks, especially in structure and dynamics, which are percolation and synchronization. So if you take note of these reports, I think it's the best literature you can read, because it's very, first of all, it's a report, so it's meant for a non-specialized audience. It's very tutorial. There are a lot of examples, and you can read as a book, for example. And then, moreover, it gives a clear indication how to move in the literature of networks, which is today very vast. As you see, if you sum up 888 citations here with all the others, you have more than collection of about 5,000-6,000 papers on complex networks. So you have a huge literature that you can use. And I leave you with a nice anecdote when we wrote this paper, which is now very famous, the first one, we ended up the first draft with 666 citations. And I said, no, it's not good number because 666 is the number of the beast. So it's the number of the evil. If you know Iron Maiden, there is also a song, The Number of the Beast, 666. So I say, no, we have to add more citation. We cannot publish a paper with 666 citations. So we made a first revision, and we ended up with 777 citations. And I said, no, it's not good because 7 is the number of deadly sins, you know. So you cannot have 3 times 7 because then people start. So we have to make a second revision. And the second revision ended up with 888. Say, okay, stop. And this we published the paper. That was very nice. So you have 888. That's exactly why I remember the number 888. Okay, thank you.